Hey everyone, I hope you're having a good day. So today's video is going to be a little bit off topic from my previous videos. There have been claims recently that CBD or marijuana can cure COVID-19, also known as the novel coronavirus, also known as the current world issue, in case you've been living under a rock, in which case you're probably safer than most of us. I wanted to make a video talking about the study in a little bit more detail than the news sources have been. So some of this information will be reheard, and some of it should be new because I actually found the paper and wanted to show everyone what they're really looking at and in what settings. I will also leave a link to the paper in the description in case you wanted to read it yourself. So, the study is named In Search of Preventative Strategies, Novel Anti-Inflammatory High CBD Cannabis Sativa Extracts Modulate ACE Expression in COVID-19 Gateway Tissues. This study took place at Canada's University of Lethbridge. It happened on April 19, 2020, or that's when the paper was published. And a TLDR for the entire thing is the novel coronavirus, currently known as COVID-19 or SARS-CoV-2. It enters the human host through a pathway called the angiotensin converting enzyme 2, also known as the ACE-2. Tested with cannabis sativa plants higher in CBD on 3D printed tissue, they found suggestions that it also worked on the TMPRSS2, which is another critical protein used for COVID-19 to enter the host, which is then suggested to be made into future things like mouthwash or throat gargle for both home and clinical use. A quick description of COVID-19 virus as we know it. Its transmission is from respiratory droplets from coughing or sneezing. However, aerosol transmission or in close contact cannot be ruled out. It was first isolated within the airway epithelial cells that were found similar to SARS-CoV, which is where it gets its name, though these cells are located in many places throughout the body, and here's a few examples. Finally, we have the ACE-2, which is a well-accepted fact that it plays a key role in the admission of the virus. The ACE-2 expression was found in lung tissue, nasal mucus, kidneys, and gastrointestinal tract. How it works is that a human cell naturally has the ACE-2 receptors. Humans have more of these receptors when our lungs are inflamed. Studies have found an increase when people smoke cigarettes, for example. So COVID-19 has spike proteins that connect easily to the ACE-2 receptors. As previously mentioned, they are using 3D printed human replica cells. The paper then goes into details about these cells, though I'm not going to. They also grew a bunch of cannabis high in CBD. They then extracted using ethyl acetate and then left stirring overnight, to which they filtered it in the morning and the concentrated version was left. Then it was tested using the HPLC, known as the High Performance Liquid Chromatography. To analyze the effects of C-sativa on the ACE-2 expression, the extracts of 22 novel C-sativa lines, which can be seen there to the right, the bolded strains on the list considered to be hemp, and the strains in red were shown to affect the ACE-2 inhibitor. Their method of testing was to create inflammation, then treat them with extracts and analyze the anti-ACE inhibitors and the anti-inflammatory properties. We will see a few different charts that better explain their findings. We can see the titles of each one will be the type of tissue being tested, and then we will see how the effect of the concentrate lowers the ACE-2 gene expression. In Figure A, we can see it was tested on oral tissue. Lines 81 and 130 reduced the levels of ACE-2 gene expression. In Figure B, they did a wider range study and found that lines 9, 45, and 167 downregulate the levels of the ACE-2 RNA. In the intestinal tissues, Figure A, line 45 decreases levels of the ACE-2 gene expression, and lines 45, 17, 129, and 169 downregulate the levels of the ACE-2 protein. Now onto something called serin protease, TMPRSS2. I don't know too much about it, and the paper details it along with the ACE-2. Serin protease, the TMPRSS2, plays an important role in the SARS-CoV-2 infection process. While the ACE-2 is the receptor for the viral entry, the TMPRSS2 primes viral spike proteins and therefore is crucial for the SARS-CoV-2 to enter the host cells. So next is what is called the discussion. It is really what they concluded more or less, though it leaves it open for discussion. The most important part, though, I thought, and I suggest you read it for yourself, was that our results lay foundation for further in-depth analysis of the effects of C. sativa on the molecular etiology and pathogenesis of COVID-19, as well as other viral diseases in which viruses use the ACE-2 receptor as a molecular gateway. Finally, their conclusion basically said the same thing, though they go on to say they believe it in the future as a preventative strategy such as mouthwash or throat gargle. Though, if you don't normally read research papers because they are boring or too long, this one is actually pretty short, being around 10 pages, and it's on a topic that a lot of people are passionate about and becoming more passionate about. Research papers are one of the best way to get first-hand information on news sources that a lot of times pick and choose what they want to be in there. Anyway, I hope you enjoyed this video, and until next time, have a great day.